Greetings everyone. In this video we're going to be talking about proteins and a variation of them called enzymes. The enzyme section won't be the most in-depth because that's going to be something a little bit more lecture focused, not so much in discussion and on these videos, but we're really going to harp on proteins because it's a very, I would say, difficult topic because it's kind of the first major overall topic that we're talking about, right? We really haven't gone over much in general, just maybe some chemistry. This is our first main part of biology, and so it can be a little daunting, and hopefully once we're done talking, you'll be a little bit more confident with how we can approach proteins, identifying them, some of their traits, functionality, all that good stuff. Okay, so in order to understand proteins, we need to actually understand what they're made of. Proteins are what we call peptides, okay? They are long strings of monomers, okay? Monomers are just kind of the smallest subunit. You put a bunch of monomers together, you get a polymer. So what is an what is a protein monomer? Well, that's going to be our amino acids. That's going to be kind of our smallest part. We put a bunch of these amino acids together and that gives us our protein. But that's a little bit further down the line. Let's zone in on what it is to be an amino acid. Now, we kind of saw a little bit of this last week or in the previous video, wherever you are. But just to kind of refresh our memories, try to label all the parts of the amino acid. I've went ahead and given you what those 4 parts are, but really you should be able to think off the top of your head what their names are and be able to identify them. So go ahead and do that. Like always, pause the video, that way you can do it yourself. I'm going to go ahead and give the answer. So the first one is the amino group, right? We're looking for the amine. That would be this group here, right? Amines are based off of nitrogen. We have the carboxylic acid group. That is here, right? It's an acid. This H is our acid, okay? So that'll be the proton that gets donated, right? That's how we define an acid. We need our variable group. Well, that's noted by R. And finally, the alpha carbon is that guy there. We won't go into more kind of crazy proteins in this class, but there are a few different types. You can have beta carbons, all the way down the Greek alphabet. And then those will have different amino acids and whatnot. But all you have to worry about in this class is the R group is going to be attached to the alpha carbon. Okay. But we kind of know what the amino group is. We know what the alpha carbon group is, we know what the alpha carbon is, and we know the carboxylic acid group, but let's kind of zone in a little bit on the variable group. What does that mean to be a variable group? And really, what we can talk about is if we only define amino acids by the backbone, right, that NCC, our NCC, well, then every single amino acid on the planet would be the exact same, right? All of these alpha amino acids, they would all have that same thing. But we know that's not the case, right? There's a bunch of amino acids out there. So how do we define what each amino acid is? And, well, that's based off of the R group. So a very specific R group will lead to a very specific amino acid. Okay, and you can get non-polar amino acids, polar amino acids, or charged amino acids, both positive and negative, based off of the R group, okay? So, important takeaway. The functionality of the amino acid is based off of the R group, okay? Because if we just said it was based off of the backbone, well, then they would be polar, right? Because we have a polar amino group and a polar carboxylic acid group. But there would be no distinctions between any amino acids. So everything is based around the R group. The backbone will come into play in one of the multiple structures we'll talk about in a little bit, but we're kind of getting ahead of ourselves here. So once again, to repeat, it's all based off of the R group. So let's zone in. I'm saying that a lot today because, we're taking an outside view and kind of going in. We're going to really zoom in on this group here, methanol, okay? It's a methyl group attached to an alcohol group, okay? Not that you're expected to memorize that. However, when we attach that in place of the R, so if we were to put our methanol right here, we would get the amino acid serine, okay? So here we have our NCC, here's our alpha carbon, here's our carboxylic acid, here's our amine, and here was that functional group, the methanol, okay? So it becomes the R group. And now we can think about, well, what sort of amino acid would this be? Well, we have a CH2, right? That's not going to do much because a hydroxyl can easily overwhelm the properties of a CH2. Now, once you get about 4 or 5 carbons in a chain, that's when we'll start to think non-polar as opposed to polar, but that's more for your organic chemistry. In this class, if you need to look at an amino acid, you'll be given a table where you can look at them, and really if you see a very polar sort of group on them, it would probably be safe to assume it's polar. Like I said, you will not be expected to memorize any amino acids or any R groups. Just be able to identify polar, non-polar, and charged. Okay, so back to this. We have a methanol, right, as our R group. So this would likely be a polar amino acid, okay? Because its R group is polar, right? Hydroxyl is a very polar functional group. And that has to do with oxygen having a very high electronegativity and hydrogen having a relatively low electronegativity. So you should be thinking back to your intro to chemistry where we talked about polar bonds and polar molecular structures, right? Okay, so to reiterate, amino acid functionality based off of the R group, that's the part that changes. But our proteins are not just made up of one amino acid, right? Because otherwise we'd only be able to have, is it 46? I think there's 46 amino acids, or 20, somewhere between 20 and 40. As you can see, I don't even remember how many amino acids there are. You don't need to know that. My point is, there's not that many compared to the amount of proteins, like the variety of proteins we need in our body. So what do? We get a bunch of amino acids together, and each unique chain makes up a different protein. And now we have so much variety. And this is really what makes, organisms differ from one another, right? They're proteins. And, well, there's quite a lot of things to that. But just for a quick glimpse into that, it does play a role. Anyways, we need to figure out a way to combine 2 amino acids together. Well, hopefully, and if you don't, that's totally okay. But if you do see it, that's good, right? You're understanding your functional groups. If you see this OH here and you have an H, well, that together makes water, right? And this carbon will lose 1 bond, right? That bond here will go away. This nitrogen will lose 1 bond. So what will happen, this will go away. This hydrogen here then bonds to oxygen. And what do you know? We have water. And what is left over? We have a carbon that needs a bond and a nitrogen that needs a bond. Lo and behold, they bond to one another. So what we're left with is a chain of amino acids bonded together by what we call a peptide bond between that carbon and the nitrogen, and water. So we will call this kind of two things interchangeably, even though they're not quite the same. In this class, you can see them being used the same thing. A condensation reaction, okay? Condensation, right, means using water, right? Well, not quite, but water is involved, right? You're condensing and water is produced. There's our production of water. Or you'll see dehydration synthesis. Okay? Meaning dehydration, you're removing a water. Here's the water we removed. Synthesis, meaning to put two things together. So a synthesis is, to make something else, right? Synthesizing something is to make something. Here we've made a peptide and we dehydrated it, right? We dehydrated it, made a peptide. And remember, this is called a peptide bond. Okay? So, we've now put it together. But what if we don't want this amino acid to be in a chain anymore? What if we want to separate them? When we eat protein, right? That's a pretty big source of our amino acids, right? We can just take it from other things. But the thing is, these amino acids are already in a protein. So we want to break them apart so that way we can use the amino acids for other things. But how do we do that? Right? So if we remove water, we make the peptide. So what could we do to break apart the peptide? Try to think about it. And hopefully what you were able to get to is if we add water, what we call hydrolysis. Okay, lysis means to cut or separate. Hydro meaning water. We're using water to separate or cut things. Okay, so if we add water, what's going to happen is our peptide bond is going to break. Let me separate all of this out. Peptide bond is going to break, and we can add our water in, right? The OH goes to the carbon to form our carboxylic acid group, and the last H goes to the nitrogen to form the amine. Just like that, we've added water to separate. So when we want to make a chain, we use a condensation or a dehydration synthesis reaction. This removes water. To separate them, we add water. So we're going to add H2O, and that'll separate the two amino acids apart. Like I said, this is called hydrolysis. So hydrolysis and then condensation / dehydration synthesis reactions are inverted, right? One breaks apart, one puts them together. It's a very important thing to know because these reactions will come up multiple times throughout the quarter. So definitely having a good understanding of how they work is going to be important. Okay, so now we've just put two amino acids together, right? Well, if we do that, let's say 1,000 times, we're going to have a pretty large protein. And we need a way of systematically categorizing that protein. So we do that based on the structures. Okay, there's going to be 4 structures. And the first one is called the primary structure. How I like to think about it is these structures go from most simple to most complex. Okay, so the primary structure, before you even know how anything interacts, you just need to know what the heck is there. So I say the primary structure is just what is physically there. And what I mean by this is it's the unique sequence of amino acids that are bonded together through peptide bonds. Okay, so this is flat out just what amino acids do you have and in what order. That's your primary structure, right? It's the most basic. You don't need to know what each amino acid, does, like is it polar, non-polar, charged, whatever, how it'll interact with the other ones, whatever. You just need to know what's there. Now remember, right, when we put amino acids together, we create that NCC backbone. N, C, C, N, C, C. When you're trying to identify proteins, look for this, okay? It's what you can also look for the amide groups, okay? That's a carbon double bonded to an oxygen, that's bonded to a nitrogen, it's bonded to other things, okay? So this here is an amide group. Notice it is not an amino group, okay? The moment you have this carbonyl here, this becomes an amide group, not an amino group. Additionally, I want to make sure this is all understood. This carboxylic acid functional group does not have an alcohol. Okay, this OH is part of the carboxylic acid. It is not a standalone. You cannot have a functional group inside of another functional group. And that's because this carbonyl affects the functionality of this alcohol. So rather than saying alcohol inside a carboxylic acid, we say it is just a carboxylic acid, just to make sure that's clear. But anyways, back to proteins, right? When we put these all together, we create that NCC backbone via peptide bonds. And the peptide bonds are within the amide functional group. Okay. So our primary structure is just what is there, right? What is the order? Like I said, how I like to explain is you can't really know how two things interact without knowing what the two things are. You know, how am I supposed to know that, I don't know, glutamine and asparagine are going to interact in some way if I don't even know that those are the two that are there, you know? So I have to know what's there before I can start looking at how they interact. But now I should be thinking, okay, I know what's there, right? It has this methionine, tryptophan, I don't know what THR stands for, glutamine, whatever. Well, THR is not glutamine, there's GLN. You don't have to memorize the amino acids, like I said. But now that you know what the order is, now we can start looking at interactions. So we should be thinking, what things can interact with one another? And remember, like I said, we want to go from most simple to most complex interactions. So our secondary structure is going to be interactions between the backbone. Okay? And this is because the backbone, right, doesn't change. You know, it's always going to be that NCC, 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 NCC. Now, whether or not it goes into an alpha helix or a beta sheet, those are the two secondary structures, is dependent on the primary structure, like what is actually there, you know, because it's all coding for this stuff. But it's interactions between the backbone through the H bonds. Remember, your amino group and your carboxylic acid group, when they combine together, they make an amide. That amide can engage in hydrogen bonding. In addition, you do have an amino group and you have a carboxylic acid group on a protein. You just have one of each. That's a part of the backbone. You can have other ones that are part of the R groups. But in the backbone, right, you can have hydrogen bonding. That hydrogen bonding makes up the secondary structure. So you went from, I know what's there. My secondary structure is what is always, like, what are the interactions between things that are always there that is not going to change. That's your NCC backbone, right? That won't change. We're assuming this is an alpha protein. So now we should be thinking, well, what the heck is the point of the R groups if our primary and secondary structures have nothing to do with the R groups, right? They're not, the R groups aren't interacting to influence those structures. Well, remember I said there are four structures: primary, secondary. Primary, what is physically there? That unique chain of amino acids. Secondary structure is hydrogen bonds of the backbone. Our tertiary structure is where the R groups come into play, right? Our R groups dictate the amino acids, and those can be nonpolar, polar, or charged. The tertiary structure is this 3D structure, right, of how these different R groups interact with one another. And there's four main ways we do this, okay? We have the hydrophilic interactions, such as hydrogen bonds, we have the hydrophobic interactions, and we also have ionic interactions. But I said there was 4. So the last one is kind of a special case. It has to do with a covalent bond. And what we say, it's a disulfide bridge. And it looks like you have an R group, or R just meaning anything, bonded to a sulfide, bonded to another, sulfur bonded to another R group. And then these R groups are part of the amino acid chains. So adjacent sulfurs can form a covalent bond between each other, making a disulfide bridge. Okay, so your tertiary structure is the interactions between the R groups. And you have hydrogen bonds, hydrophobic interactions, ionic bonds, and disulfide bridges. And really quickly, what sort of interaction is depicted here? Well, let's see. CH3 is a methyl, and we have another CH3, which is another methyl. So we have 2 methyls interacting with one another. Are they polar or nonpolar? Well, it's just carbon and hydrogen. We said that's pretty nonpolar, so they're going to be 2 nonpolar functional groups, R groups, and so they're going to engage in hydrophobic interactions. This is just one example. Okay? So, primary structure, unique chain of amino acids, right? The unique sequence. Secondary structure, hydrogen bonds between the backbone. Tertiary structure, interactions between the R groups via H bonds, hydrophobic interactions, ionic bonds, and disulfide bridges. So what is the 4th one? Well, the first 3 are all within a peptide, meaning one chain of amino acids. But could it be possible that a protein consists of multiple peptides? It actually is. So a protein could be made up of a single peptide, but it could be made up of multiple peptides, each making up a different subunit of the protein. So now we have our 1 through 3 structures. They are within a peptide. Our quaternary structure, the 4th structure, is between peptides. So meaning we have peptide 1 and we have peptide 2. They are going to interact in different ways based on their shapes and their R groups, right? And so it's labeled as van der Waals forces here, but really we're going to say that's hydrophobic interactions. We have our ionic bonds. We have our covalent bonds, which include our disulfide bridge, and we have our hydrogen bonds. Okay, so primary, secondary, and tertiary within a peptide, quaternary structure. Our protein is so big, so complex that it needs multiple peptides. How are they going to interact with each other? We will label that as the quaternary structure. Okay. Quick practice problem. How many amino acids are depicted in this peptide? There's a few different ways to go about it. Choose which way works for best for you. I also want you guys to label the N and the C terminus. As always, stop the video. I'm going to go ahead and assume you have. And let's talk about the answer. So, first, let's label the N and C terminus. We're looking for an amino group. It's pretty convenient. Here is our N terminus, right? Our amine is the furthest out. And here we have our C terminus because it's the carboxylic acid. Okay, but now how many amino acids are depicted? Well, so you could count how many NCC repeatings you have. However, it's pretty easy when it's in a straight chain like this. What if it were kind of all over the place, right? It'd be kind of difficult to track. You know, maybe it's straight and then it goes up with a side chain like that. So it's not always easy to do that. Okay, we could also count the R groups, right? Because every amino acid has one R group. So if you have 12 R groups, you must have 12 amino acids. You could do that, except you'd have to be able to memorize every single R group and be able to identify them. It's just not always easy. So the way I like to do it is I like to search for the, what's it called? I'm totally blanking on the word. The amides, right? And I actually realized I have not drawn in everything. That's not good. There should be an H there. Let me do this. I don't know how I forgot about that. Okay, I'm going to fix this and I will be right back. Okay, I apologize for that. I totally slipped my mind to fill in the other hydrogens. Because honestly, I do bond line formula so much that I think in terms of bond line rather than Kekuli. But anyways, so let's zone in on the peptide bonds, right? Because the amount of peptide bonds we have, if we add one, will give us the amount of amino acids, right? Because if you have 2 amino acids, 1 peptide bond, so number of peptide bonds plus 1 is equal number of amino acids. Okay? That's the way I personally like to do it, because that way you're understanding the different functional groups that are present. Okay, so we have an NCC. Here's our first amide, right? And so our peptide bond is going to be right here. Okay, our second one, right here, peptide bond. Our third one, here, peptide bond. Fourth one, or wait, 1, 2, 3, no, I was right. Fourth one, here, for peptide bond. Remember, the peptide bond is always between the nitrogen and the carbon that is part of a carbonyl, meaning it's a carbon double bonded to oxygen. Had a lot of questions of, for instance, on this guy, is it this the peptide bond or is this the peptide bond? Well, this can't be because this carbon is not attached to a carbonyl, or it's not a part of a carbonyl. It's not double bonded to an oxygen. So that means it's not part of a different amino acid. It's part of the same one. That's the alpha carbon. Okay? This one, on the other hand, you have a carbon that's part of a carbonyl because it's double bonded to an oxygen. That means that was once a carboxylic acid of a different amino acid. There's our peptide bond. Okay? So we have our peptide bond and another amide. So we have 1, 2, 3, 4, 5 peptide bonds, 5 amides. That's going to mean we have 6 amino acids. And if you want, we could count the number of nitrogens. We have 1, 2, 3, 4, 5, 6 nitrogens that are part of the backbone. So really, either way that gets you, or any of the ways that gets you to the right answer works just fine. This is my personal way of doing it. Finding the amides, finding the peptide bonds, and realizing you just count the number of peptide bonds, add 1, and that gets you the number of amino acids. Okay? So now I want to do a little kind of divergence to talking about enzymes, because they're a pretty important, I mean, all proteins are very important, but they're one we'll kind of talk a little bit about on more in-depthly than just general proteins. I know when I took this class, my professor, I think, actually studies enzymes in his lab, and so we did a lot of work on enzymes, and I believe you guys will do a little bit in depth too. So I wanted to include this for you guys as extra help. So let's talk about the function of enzymes. Okay, all enzymes are in fact proteins, but not all proteins are enzymes. Okay, you have proteins that do other things, right? They're not just all enzymes. So that's kind of like, you know, all rectangles are squares, but not all squares are rectangles. That sort of thing, right? Anyways, so let's talk about their function. Enzymes really act as biological catalysts, and they speed up reactions, which tends to be a little bit of a misleading term I found with students, but bear with me. So it speeds up reactions by lowering the activation energy. So essentially, in order to get a reaction to occur, the molecules have to touch and you have to supply enough energy for the bonds to break. Okay? Catalysts, these biological catalysts that are enzymes, can help lower the activation energy, meaning you don't have to supply as much energy in order to get the reaction to go. That's how they speed up, quotation marks, the reaction. So let's talk a little bit about the structure. Enzymes have an active site. This is physically where the reaction takes place. This is where the enzyme binds to the substrate or the substrates. And then you also have the allosteric site. This is more on the back side where regulatory molecules can bind. So this is not where the substrate binds. This is where regulatory molecules bind, okay? Now, you can catalyze a reaction in 4 ways. You can position the substrate in optimal orientations, okay? So meaning that the substrates are going to come together in a way that promotes a reaction. And this is all very conceptual, think in your mind like a, positioning things around. Okay. It can also compromise the bonds so that they can be more easily broken. The way I think about it is, if you're ever really struggling to, tear something apart, you can twist it, right? If you twist it, like think about like putty, you're trying to separate the putty. If you twist it, and you twist it and twist it and twist it and twist it in opposite directions, right? The 2 sides, opposite directions, it'll make it more easily to break apart. Same thing that's happening here. Okay? The enzymes can also, for third thing, provide a micro environment for a reaction, meaning you have it occur at a specific pH or temperature. So essentially in this active site, let's say this is the active site, you might have a pH of 9, whereas the rest of the cell you might have like a pH of 4. Okay, maybe this reaction really needs a pH of 9 in order to go, and we'll talk about why that is in a future video when we start talking about pH versus pKa. But anyway, so it can provide a microenvironment, a specific pH or specific temperature, so that reaction can occur in optimal conditions, but perhaps are not conditions that the rest of the cell would like. Or they can actually take part directly by forming bonds with the substrate. So they can, physically form covalent, ionic bonds, whatever, with the substrate and take part in the reaction. There's one very important thing though. In order to be a catalyst, you must be both a product and a reactant. Meaning whatever state you went into the reaction as, you must come out in that exact same state. So, if you form a bond with the substrate, in order to be categorized as an enzyme or a catalyst, that bond must be broken and you must return to your initial state. Otherwise, you become a reactant, and whatever you turn into, that's not what you were, is now a product. Okay? Especially in, I think, Chem 2C, they love to put that kind of question on a test. If you've taken Chem 2C, of how do you define a catalyst, and it must be both a product and a reactant. So essentially, it must be unchanged, right? It can go through minor changes, but those changes must be reversed by the end of the reaction. Okay. Enzymes also can be regulated to ensure proper levels of substrates and products are maintained in the cell. So meaning, how I like to explain it, okay? If you are just making a bunch of spaghetti, I mean, we're all college students, we like to eat pasta. I mean, I live off of pasta, I won't lie. And you just keep making pasta, right? Just keep making it, keep making it, keep making it, keep making it. It doesn't matter how much is in your kitchen, you just keep making pasta. Eventually, you're gonna run out of place to put all this pasta, and it's gonna go to waste. Okay, if you just have your enzymes going and 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 going, you're going to run out of your substrates and just have a bunch of excess products that are not being used. And now you've just wasted all that energy making products you're not going to use when you could have been doing other things with your energy, like movement, or maybe you needed to do another pathway that required energy. So we can regulate our enzymes both through competitive inhibition, meaning that a substrate and a inhibitor both can go into the active site. The inhibitor can block access to the enzyme, or block the substrate's access to the enzyme, or you can work through allosteric regulation, meaning that a regulatory molecule will bind to the backside of the enzyme to alter its function. Now, quick misconception is this must always be inhibition. Not necessarily. A protein can be off by default, and only with the presence of a regulatory molecule does it become active. Now, we're going to get into a lot of that later on, especially when we talk about like gene regulation. This really comes into play, regulation. But probably a little bit now you'll talk about it. So just to give you guys a heads up, there is regulation that takes place, and it's not just in the active site, it's also in the allosteric site. So like I said, the two ways a protein can be off by default and it's turned on by a allosteric molecule, or it can be on by default and turned off. So essentially we have a molecule bind to the backside of the protein, and then all of a sudden this becomes deformed, and now its shape no longer matches that of the substrate. So there's kind of multiple ways you can go about it. Most importantly though, enzymes do not change the delta G of a reaction. Okay? They lower the activation energy, but they do not change the Gibbs free energy of the reaction. So if we draw it out, okay, let's say here is without an enzyme. You should be thinking an exothermic reaction, right? Because the reactants have a higher energy than the product, so energy was released. Okay, and then with an enzyme, it might be down there. Whoops, perfect. Okay, so with enzyme, notice how the E sub A is a lot smaller, okay? It lowered the activation energy. However, the difference, right, the delta G between the products and the reactants is the exact same. It does not change the delta G of a reaction. It only lowers the activation energy, okay? So that is really important. Professors like to, on like the first midterm, throw in a question like this about enzymes. And it's designed for you to forget about what they do, because we get in this mindset of, oh, they lower the activation energy, they change the amount of energy we get out of a reaction. No, it does not change the delta G. It only changes the activation energy. Okay? So that is really important to understand. All right. Have a few more things I want to talk about. Like I said, enzymes are proteins with a specific sequence of amino acids, especially in the active site. Okay, so right, they're proteins, right? They have a very specific sequence of amino acids. But I want to highlight the active site. If you have a bunch of non-polar amino acids, right, in your R group, is it likely you're going to be reacting with a polar substrate? Probably not, because they're going to want to repel each other. And remember, our tertiary structure is going to have its 3D shape, right? So if the 3D shape is not compatible with the substrate, which is dependent, right, on the R groups, well, then it's not going to bind, right? You have to have the right enzyme with the right substrate. Think about it, like that, and then you have it around, right? If you were trying to fit a pentagon in a active site that looked like that, probably wouldn't work. But if the active site maybe looked like that, maybe it will work. So only a specific substrate can interact with a specific enzyme. This is what we call enzyme specificity, okay? And this has to do with the enzyme, right? The active site of the enzyme and the substrate being polar, nonpolar, charged, small, large, whatever quality you would like to give it. It's all based on those qualities. However, I should note that you can use multiple substrates, okay? Sometimes compounds can look very similar to one another. They have all the right things in all the right places, and it can bind to the enzyme. You can also have multiple substrates using the same enzyme at the same time, right? Maybe you have one guy here, and you have another one here. And maybe this enzyme is catalyzing the reaction between these two, okay? So there's a few different variations we can have with enzymes and substrates. But this is so important that I wrote it twice. Enzymes always return to their original state after the reaction has occurred. Actually, I didn't write it twice, stated it before. They must always be in the same state as they were at the beginning as they are at the end, okay? If they are altered during the reaction, they must be altered back. They will be in their original state after a reaction has occurred. They will be both a product and a reactant. Okay? It's very, very important. If they are altered in any way, they will likely be more of a reactant, not a enzyme or a catalyst. Okay? So, a few important things to understand with enzymes and catalysts, right? Returning to their original state. Don't alter the delta G, can do all these things, but they are, critical for maintaining life, and that's why we often use them to kind of give you an example of proteins. Now, enzymes are also really, really complex things, and so we're giving you definitely a simplified view, but it's I think it's helpful to know this, at least in this simplified view. Okay, kind of our last thing, I guess two things. I want to talk about environmental conditions. So, a protein structure can actually change depending on the temperature and pH of their environment. So, if we change the temperature, right, as we increase the temperature, the molecules themselves actually start moving faster and faster. And when things start moving faster and faster, reactions tend to happen more quickly. But what if you add so much energy, right? Increase the temperature so much that all of a sudden the bonds that are keeping the structure together start to break. Because remember, the bonds only contain so much energy, and once you supply enough energy to break those bonds, the bonds will break. That's what's happening at this cusp here. You've now supplied so much energy that the bonds that are holding the protein together are no longer able to keep the structure. And all of a sudden, what we say is the protein denatures, or the enzyme denatures, meaning that the structure of the protein changes that leads to an irreversible change in their function. I think they might be able to be, in some instances, be reversed. But in all of my notes that I have, it says an irreversible change in function if you denature it. So I'd be wary of that. And so, this will affect the secondary, the tertiary, and the quaternary structure because they all have to do with the bonds keeping that 3D structure together. Now, the same thing works with pH. If you increase the pH to a certain extent, the the reaction rate will increase because, well, we'll get into that a little bit more in the pH versus pKa video. But how it was taught to me is using this diagram here. So right when the pH is equal to the pKa, you're going to have about 50% of the protein being protonated and 50% being deprotonated. If you need a refresher on that, you can check out the Intro to Chemistry video. When the pH is less than the pKa, you're more likely to find the protein protonated. And if the pH is greater than the pKa, you're more likely to find the protein deprotonated. So if you require this H here, right, in order to hydrogen bond as an enzyme, and all of a sudden you crank the pH to above your pKa, you're probably not going to have that H, and all of a sudden you can't use that H for hydrogen bonding, and now you won't react well with your substrate. That's what's happening here. It could also happen the other way. Maybe you really don't want that hydrogen there. You want to be negatively charged in order to interact. Well, if you lower the pH, you're going to attract that hydrogen to you, and all of a sudden you don't have that negative charge to interact with your substrate. So pH and temperature both will change a protein's structure, and it can become an irreversible change, and that's what we refer to as denaturing the protein. Okay? And there will be an entire video on pH versus pKa because I found it is one of the most difficult topics for students to understand. In fact, last quarter, the professors didn't even go over pH versus pKa. My professor did, and I believe your professor will. I could be wrong with that, but I will go ahead and make a video on it anyways, just so you guys have that resource. So the final thing we want to talk about is enzyme regulation. Okay, I'd mentioned this very briefly earlier, but I want to give a little bit more time with it. We have competitive inhibition, meaning that the substrates compete for space in the active site. Remember, the site that is actually working with that the substrate of that enzyme, okay? So you have an enzyme active site, and you have one substrate in here. Well, if that substrate's there, this substrate can't get in there, okay? So these two are competing for this enzyme's active site. That is competitive inhibition. And so now, rather than using another substrate, what if you used an inhibitor, right? So an inhibitor looks exactly like, well, maybe not exactly like, but looks close enough. You squint really hard and you're like, yeah, okay. But it looks close enough to the substrate, and it binds to the enzyme, and all of a sudden nothing happens, right? No reaction takes place because that inhibitor is not going to want to react with anything. But the substrates that would have react now can't because there's something blocking you, right? It'd be like, trying to park in a parking lot or in a parking space, but there's already a car there. It's like, well, crap, I got to go drive around for 20 minutes trying to find another parking space. That's competitive inhibition. Okay, but we also have, like I mentioned earlier, allosteric inhibition. And these are molecules that bind to the allosteric site, altering the function of the enzyme. So here we have our active site, and this green guy is our allosteric regulator. It doesn't always have to be inhibition. I should probably make a note of that. Inhibition or activation. So really, it's just allosteric regulation. That's really the better term. My apologies for that. But anyways, so when this molecule binds here, it'll change the shape of the protein. And when you change the shape of the protein, you change the shape of the active site. So that could make the active site into its proper shape, right? That would be an activation. Or you can change the structure of the active site so that it can no longer fit its substrate. That would be inhibition, okay? So we have competitive, that relies solely on the active site. Allosteric deals with the active site, which is not, Allosteric regulation deals with the allosteric site, which is not the active site. It's so annoying that they all start with A's, man. Come on, my brain can't keep up. But I digress. And then our kind of third method of enzyme regulation is compartmentalization. So if you remember back to, or actually you guys haven't learned about this, but hopefully you will, kind of the cell structure. Maybe you guys have gone over it. I can't remember. My brain is fried. So in eukaryotic cells, enzymes are separated by membranes into different areas to concentrate the same processes in the same area. So here I have like an example of a cell, not really special or anything. This is probably not realistic at all. But as you can see, we have all these folds in the membrane. And what do we have? We have all the pink enzymes in one area. We have all the green enzymes in the other, and all the lime green enzymes in the third. So we compartmentalized it. Remember, some of these things require a very specific pH or a very specific temperature to operate, to have, ideal reactions. So by compartmentalizing, oh my God, words are so annoying. When you compartmentalize something, you can make it so that they have their each sort of like microbiome of sorts, microclimate, whatever you want to call it. Just an area with a very specific pH and temperature so that the enzymes can operate at, whatever efficiency you need them to. So by kind of by sectioning off areas of the cell membrane, you can concentrate all the resources you need in one area and have optimal conditions for that. And that's another way you can regulate enzyme activity. So you wouldn't have a green enzyme way the heck out here because this area maybe it doesn't have the resources, it doesn't have the substrates it needs. Maybe out here is also suboptimal conditions. So you put it all right here, you supply it with all the substrates here, and you make sure you have the optimal conditions to sustain your reactions here. So those are our different ways of regulating enzymes. Those are the, major ones in my opinion. Like I said, these videos are not meant to be a complete overarching overview of everything in the class. They're really supposed to be, maybe you need just gaps in your knowledge of the very main ideas. That's the goal of these videos. Because, if I wanted to teach you guys everything in this class, I'd be here for days trying to do it because that's what your professors are doing. And plus, your professors are a much better source of the information than me, right? Because your professors are the ones writing your tests. So best to learn it from them and have me just kind of, help you out, supplement your knowledge with kind of some of the main ideas. Especially, when it comes to final season and you're like, I can't remember what I talked about on the very first day. I just need the main idea and that'll hopefully jog my memory. That's the sort of thing I'm getting at with these videos. So these are not, like I said, not meant to supplement going to discussion or lecture. Go to both of them. These are just supplementary videos to assist in your learning. As always, I hope your studies are going amazing. If you have any questions, please do not hesitate to reach out to me. My goal is to help you guys, help yourselves as students. I have confidence in every single one of you. And I know you're going to do great in Biz 2A and great in all your other classes. I'm proud of you guys for sticking with the program here. That is all. Goodbye.